Thank you, guys. Well, I'll get started here. So I did this project back in summer of 2011, summer of 2012. So it's been a few years ago. But we actually just got the paper published in 2017, I think after our fourth revision. So I'd say for any grad student in here, if you're working on getting something published, don't give up. Eventually they'll give in and let you publish it. So, but what we were working on is Goss's wilt had kind of started to become a big problem again, especially in Nebraska. Kind of trying to see, like, why did it reemerge and how can we get a lot of information from across the whole state as well as Iowa to kind of see if we can figure out factors that were kind of contributing to that reemergence of the disease. So kind of a little introduction on it, as corn or maize is an important crop in the U.S. 89.1 million acres were planted last year, or 36 million hectares. It's grown in all 50 states. And we exported 2.45 billion bushels, or 62 million metric tons of corn. So for the U.S. economy, corn's a really big deal, especially for the grain farmers in the U.S. Makes up about half the acreage of farmable land that we have. The top 5 corn-producing states in order: Iowa, Illinois, Nebraska, Minnesota, Indiana. Something else all these states have in common: they've all had Goss's wilt in the past. So it's out there, something we have seen. Kind of a background on the pathogen. So where this was first found, you see that right there, Lexington, Nebraska. That'd be right on I-80 in central Nebraska, probably under center pivot irrigation out there. The name of the pathogen, Clavibacter michiganensis, subspecies nebraskensis. We'll kind of labeled as CMN for short. But as I said, first occurred in Dawson County, Nebraska, out there in central Nebraska in 1969. Kind of the spread of the disease by 1970, they found it in 9 counties. By 1971, it was 16 counties in Nebraska and in Iowa, and then just kind of continued to move. We saw it in a lot of counties. I think by 1979, they were up to 54 counties out of 93 in Nebraska, so over half the counties had this disease in it. And then after 1980, it was fairly sporadic. It was kind of a real big deal there for a few years. I think they did some breeding, found some tolerant or resistant or tolerant products to the disease, and really didn't see a big issue there. But then in about 2006, Tamara Jackson said they started to get a lot of calls out of there at Panhandle, Nebraska, southeast Wyoming, and northeast Colorado. We're starting to get a lot of calls that they're starting to see Goss's wilt reappear. And I was talking to Jim here, this morning a little bit about some of these epidemics in the past, and I think it was the same thing here. These seed companies hadn't seen Goss's wilt since the early 1980s. So in 2006, nobody was looking at, do we need to be breeding against this pathogen or screening? I think in 2006, it said 25% of seed companies had a rating for Goss's wilt in their seed catalog. By the time I did my survey, they were back up to 90. So people were the companies were starting to say, hey, we need to take a look at this again. We need to at least have ratings, have screening programs out there to make sure we keep up on it. Current distribution. So by 2008, Nebraska, Illinois, Iowa, South Dakota, Minnesota, Wyoming, Colorado, Kansas, and Indiana had it. And then some of the newer reports: Texas in 2009, Canada 2010, North Dakota, Missouri, and Louisiana. So pretty well spread across the U.S. I'm guessing you can probably find it in a lot of states that are in the Corn Belt. They just haven't confirmed it officially yet, but pretty widespread. So a little bit of background on the pathogen itself. So it overwinters in infected plant residues. So like a lot of our diseases, it's one it's going to be in the field next year until that tissue breaks down. That pathogen is going to be there. I know did some studies they did back in the 70s when they first found it. They buried the residue. Said after about 10 months, they would find it still living at 10 months. After that, if the residue was broke down enough, they wouldn't be able to get that pathogen back. Another thing, it can be seed transmitted, very low percentages, but that could be another point of dissemination for it. Typically, where we see this, and this is why I think it's especially bad in western Nebraska, we get a lot of hail. Opens that plant up, we get high winds. Certain parts of the state, sandblasting is a big issue, where we get 70-mile-an-hour winds on freshly tilled sand, and it'll just eat into those plants. And even hard rain can punch holes in the leaves. So most of the time when we see this pathogen, most of the research in the past has been done saying that it needs physical wounding. I know Allison Robertson at Iowa State has had a student working on, can it get in through the stomata, can it get in through The droplets of water on the leaves overnight, the guttation droplets, can it get in that way? I think they found in some cases they were saying that it could, but I think most of the time we're still seeing it through physical wounding events. 2 phases to the disease. So the top one up there is the leaf blight phase. So I'd say 99% of our cases are this leaf blight. Why we're losing yield is because we're losing photosynthetic area on the plant. But if we get a real bad field, especially in those sandblasting areas, you see that bottom plant there is the vascular wilt, which is a pretty tough one to deal with. It'll typically kill the seedling. This just kind of shows a few more of the leaf blight phase. So what you kind of look for on that, things we'll see are kind of these water-soaked, or these little, they call them freckles, but they're kind of a water-soaked spot. You kind of see that shiny appearance. It's not real easy to see here, but it's almost like an orange exudate that bacteria is pushing out of the cells of the plant that you'll see on the leaf surface. And those are kind of the main characteristics of it that we kind of visualize in the field. I'll get into a little bit more of the lab-based stuff we did to confirm it. The other one's going to be the vascular wilt stage. So this is what we call a V11 growth stage, basically 11 leaf collars. That'd be probably a corn plant that's a little bit taller than me. If you look at those plants in the middle that have the vascular wilt, they're about knee high on me. So as you can see, any plant that has that is probably not going to be a productive plant. It's just going to be a weed out there in that field for the year. And kind of what's happening with those in the vascular wilt, we're actually getting bacteria in the vascular bundles that is not allowing water nutrients to go up into that plant, and that's why it stunts its growth. But you kind of look at the size difference from the picture in the bottom left here. Hit the wrong button. But that one there is about 1 inch. This would be a plant that's in the same field where the vascular bundles have been blocked up for a certain amount of time, probably down to about 1/2 to 3/4 of an inch. And you can kind of see up here, it's just the tissue just starts to eventually rot out with that gases or the bacterium in the stalk. So pretty hard on the stand if we do get it. Like I said, the vascular wilt I haven't seen a lot of cases of. Very susceptible products, and we usually have to have the hail or that sandblasting to go with it for it to be that bad. But typically that's going to be in corn that's probably 4 to 8 inches tall. Once the plants get bigger, they seem to be able to hold it off a little bit. We see more of the leaf blight phase. So kind of why are we concerned, this disease in general? Well, the thing is, for yield losses on this one can be huge. Some early studies showed a 44% yield loss. Current observations showing 50 to 75%. Just talking to farmers, maybe they exaggerate a little bit. But a guy said, my yield goes 200 bushels. Variety A made 210, variety B made 130. Side by side like this, where the Goss's wilt was so much worse than the one that wasn't resistant. And you can see that here. Look at this on the left. We have 95% of our photosynthetic area yet when that plant's tasseling viable. Versus one on the right where we've probably lost 50% of our tissue. That's really going to drag our yield down. So that's kind of the big deal with this disease, is if we get a susceptible line of, or susceptible product, it's very hard on our yield. So current management, kind of before we started our research, what we were telling everybody, resistant hybrids are going to be the biggest thing you can do. Talk to your seed dealer, talk to your whoever you're getting seed from company. Make sure you're putting something out there. If you've had Goss's wilt in the past, it's residue borne, it's going to be there again. Using tillage to help decompose residue. There's certain places that probably shouldn't be tilled, but there's other places, if we can get away with it, it's good to bury that, help it break down faster. Rotate crop to a non-host. So for Nebraska, we grow a lot of soybeans. You can just go to soybeans for 1 year, get your corn residue broke down. It would probably help quite a bit. Reduce alternate host populations. So there's a lot of other weeds that are hosts. Actually, grain sorghum is a host. Barnyard grass. I know several of the foxtail species. So a lot of the weeds we have in our soybean fields, we would need to make sure we kept down pretty well. Then chemical control. There are some bactericides on the market, but they're really, I think, kind of pushed towards the fruit and vegetable industry because of the cost. To put them on $3.50 a bushel corn, it's just not going to pay the way it does on a higher priced, food item. So no real good chemical control at the moment. The other issues we kind of saw out there was that the trait that controls resistance is quantitative. Additive gene action is important for inheritance of resistance, and there is no true immunity. So there's not a product out there that won't get it. It's just to what severity are we going to see it. The other problem we have is ratings aren't universal. Monsanto and DuPont sue each other about twice a year over something, so they're not going to share their rating scales with each other. So, what Monsanto might call something that's really good against it, maybe pretty average on the DuPont scale, but we did our best to try to take ratings from each of these companies and kind of average them out in the project. But that's another issue the industry has in general, is there is no just, it's not like a university doing all the screening for everybody's products and giving you a list. They're all pretty in-house, and they don't want that out there, so that makes it a little bit tougher. So for this portion of the project, this was kind of our objective to identify and prioritize the environmental and agronomic factors contributing to Goss's development. In cornfields, as I was telling Jim earlier, I said the fortunate thing for me was the year we did this, the Goss's wilt was huge. In 2011, it was everywhere, places they had never seen it before, which was bad for the farmer, but very good for my project, because we got a lot of responses back. And like I said, since then, I don't think you'd had a real good year to do it again. I just haven't seen, even in my work, I haven't seen a lot of it around, which is a good thing, but at the same time, it's wouldn't make it tough to do this. So on the survey, what we did, we kind of put a survey, I believe we had 16 questions that had a checkbox. We got a very good response on that, which is probably more the agronomic ones. When we got more into the pest and disease pressure, where you left to fill in the blank on a survey, responses were pretty average. I'd say, sometimes out of the 486 surveys we got back, in some cases we may have only gotten 100 responses on some of those open-ended questions, but if you had a checkbox where all you had to do was exit out, we got very good responses. But, agronomic practices, things people can do at the farm level, rotation, population, planting date, yield history, residue cover, their tillage, irrigation, if they're irrigating, what were they using, nutrient applications, were they doing everything up front, pre, were they doing things in season, hybrid selection, was that a big factor for them, manure application, site characteristics, in these fields where we were seeing this disease this year, did they have some severe wind that maybe they wouldn't have typically seen, did they have hail where maybe they hadn't seen it before, has this disease been in the history in that field? Do they know they had it? They went ahead and planted corn anyway. Got some latitude and longitude, and then growth stage. And then, as I said, a few more fungicide, herbicide, insect, and weed pressure questions. We distributed a kit, trying to make it easy. So basically on the bottom right there, we put together a letter, kind of stating what we were doing, why we were doing it, trying to kind of get the farmer to, see why we're doing this. We're not trying to make an example out of it. We just wanted this information. It's all anonymous. There's a survey up there. And then put a Ziploc bag to send back samples in with a self-addressed postage-paid mailing envelope. And we sent out about 2,400 of these between Nebraska, Iowa State. I think we had 7 other states that had at least a few samples, but the majority were Nebraska and Iowa State. Requested a completed survey, 4 to 5 leaf sample. And what we really wanted was people to do was, if they had a field they thought they had Gosses in, we wanted that leaf, but we also wanted them to find a field where they didn't have Gosses, send us that leaf. We definitely got a lot more samples where everybody sent them because they thought they had Gosses, but fortunately about a third of them turned out negative for us. So, and the incentive on it was a free Goss's wilt testing, which in our lab at Nebraska would cost $10 at the time. So for guys that sent them in, if it was free, we'd test for them, let them know what they had. So the testing procedure on this one, basically what we do is you take that leaf out of the bag, set it on a paper towel, take a razor blade, cut a nice chunk of the lesion out, use one of these Agdia test kits here, and you can go ahead and kind of grind up, mash up that corn leaf in the solution that was in there. Take that little bit of liquid out that had some of the bacterium in it, pour it into a test tube, and then we put the Agdia test strip right in there. And basically how those little strips work, not a lot to them, so 2 things I'll say on this one. First, it basically looks just like a pregnancy test. You get 2 pink lines, that's a positive, 1 pink line's a negative. The other thing is, this test is actually made for Clavibacter michiganensis, subspecies michiganensis, which is a tomato wilt. But it works on both. The student that was in Tamara's lab before me had done the research to see what they could use this. Found that it worked very well. But that's what they do. You set them in that solution for 30 minutes, you leave it sit, you come back, and it'll either has 2 lines or 1 line. To kind of test to see if we had some that were I guess if they were symptomatic or asymptomatic, we'd go ahead and do bacterial screening on them. So if I had a leaf that I thought, hey, that looks like northern corn leaf blight, but it came back positive on the idea strip, we'd go ahead and check for bacterial streaming. And the same thing the other way. If a leaf really looked visually like it had Goss's wilt, but came back negative, we'd do a bacterial streaming check on that as well, so under the microscope. So kind of our catch to make sure we got everything correct in there. So this is kind of getting into the results of what we found. So like I said, we got 486 of these of the 2,400 we sent out. We got 486 back. 340 of those tested positive, 186 tested negative. So right at 70% of our samples were positive, which turned out to be good for kind of running some of the analysis we did on it. But of those results, we got, I said Nebraska and Iowa were kind of collaborating between Tamara and Allison and Robertson on this. I think we got like 44% back from Iowa and about 42% in Nebraska, with pretty good, varying amounts of what was negative to positive, and then as well as we got some back from Colorado, Indiana, Kansas, Minnesota, Missouri, and South Dakota. So had several states participate. I think Illinois was the only state we sent samples or sent bags out to. We didn't get anything back from. So pretty diverse set of things here. I guess the other thing on that survey, as I said, the front page was filled out real well. Some of the questions were, pretty average number of responses. So if we didn't have at least 50% of the information, so that'd be 243 surveys with a question, we kind of ran that data through a chi-square analysis to see what we could find out. And like I said, most of those were questions that had too few responses for overall analysis. The advantages to, so this would be the other parts of this, which if anybody's familiar with statistics, this is definitely not my forte, but I'll do my best to explain the programs we did use. I had a very good statistician on my committee that helped me out a lot here. But CART is basically, it's a useful prediction tool and classification. It takes a complex data set. So you're talking 486 surveys with 40 variables, and it's basically putting in order of importance which variable is causing Goss's wilt, or most likely to be causing the Goss's wilt. It handles missing data well. They said if you would test it, if you get the right answer from the producer, 80% of the time it's going to put based on other surveys we're getting back, it's going to match that data up, and 80% of the time it'll have the answer that you would have gotten from that guy. Disadvantages of this program, it's relatively unstable, and results vary every time you run the program. The results are a little bit different. And we'll kind of get into that when I show some of the graphics. I can explain some of that a little bit better. To kind of combat that, we went ahead and used random forest. So what random forest is doing is CART's making basically what they call a decision tree. But it's running like 2,000 of those trees, and it's averaging them all and putting them on a graph for you. So again, very easy to understand. This one also handles missing data well. And the measurements will be based on a mean decreased Gini plot, which I'll kind of show you that too, but just the higher the score, the more important the variable was to the development of this disease. So we'll kind of start out with these chi-squares, ones where we were a little bit short on data. We asked producers, in these fields, were other diseases present? Do we think that maybe if we have a gray leaf spot or something, is there more disease because that plant's a little bit weakened, or are we seeing these in conjunction with other diseases? Really didn't find any diseases that really came, any other disease that showed up as a match for it. So, and that was out of about 327 responses out of 486 on the survey. The first one we saw something back on was the root feeding insects and leaf feeding insects. So in most of these cases, we also asked what these insects were. In most cases, we're probably looking at, probably corn rootworm in a lot of these cases is probably the biggest pest we see in those two states for leaf feeding, and we also asked what insects, but didn't get, got more response on this and less response on that. We kind of asked about the corn feeding insects, which specific ones. I know there's also grasshoppers we asked on, didn't get very good or just didn't have enough data points, but of the ones we did, corn rootworm came back that there was an association with the feeding of corn rootworm on that. Alternate hosts in the field, so we went through their data. Guys said I had this, samber, I had giant foxtail, I had barnyard grass, and I basically grouped them in either yes they identified an alternate host to the pest was in that field the year before, or no, it was not. and didn't see any correlation with any of those weed species being a problem with seeing it again. Using a foliar fungicide. Now this is one we found to be kind of interesting. Did find a positive correlation when a foliar fungicide was put on. This kind of set up the basis for the grad student to follow me up. And then the other one we found out that was interesting was that we asked what what guys were using for herbicides. And of the 4 most commonly used herbicides, we found glyphosate to have a correlation to Goss's wilt development. And again, this was a student. I'll kind of touch on that in a little bit as well. And then the last one we did was on nutrient application. Again, didn't see any correlations to any of these nutrient applications and Goss's wilt in the field. So kind of our 5 factors related to Goss's wilt development we saw from that was that, insects feeding on the corn root system, insects feeding on leaves, corn rootworm feeding, and a foliar fungicide application. I would say most of these have a few things in common. One is when we have insect feeding, we're going to have a wounding event in the plant, so that's probably going to help out to get the pathogen into the plant. Another one for these 4 factors is good chance we're in a corn-on-corn situation, and since the pathogen is residue-borne, probably more likely to see these 4 things happen. Now the glyphosate application kind of stuck out. Talked to Allison a little bit on it, and they kind of formulated that, probably just the amount of people using glyphosate now would be a part of that. But they actually did do a research project. Sarah Schlund did this in 13 and 14. They went ahead and did greenhouse and in-field trials, doing basically her whole research project was foliar fungicide applications, glyphosate applications, and adjuvants. And they found no increase in disease prevalence in those studies. So that was kind of what I said based off my whole project. They got another student to do that, and they didn't find anything in that. So I guess maybe it was a dead end, but we thought it was interesting seeing that from ours. So then I'll kind of get in. Here's the main results. So this is what that CARP program produces. So we call this, they basically call it a tree. So you're looking at the top being the root, where you've got, up here there's our 486 surveys total. And we're trying to decide from that top point, which factor is the most important in driving whether or not we have Gosses in a field. So I'll come back to this. But, these are the ones we're saying, least likely is on the left, most likely on the right, and why is that the case? So for our population, greater than 67,500 seeds per hectare, 28,000 seeds per acre, basically. Probably in Nebraska, we're looking at irrigation, Iowa, pretty standard population. But when we get a little bit thinner, we've got 29 negative surveys and 11 positive surveys. So we're saying if you had a lower plant population, probably less plant-to-plant competition. I think there was another study done in 2015 where they showed the plants, if you infected a single plant with Goss's wilt, any plant surrounding that was touching that plant would also be infected fairly quickly after. So thinking that the reason we weren't seeing this as bad in these lower populations is probably because we don't have the plants touching each other, rubbing on each other, spraying the pathogen down the row and across the row. Was a big reason for that. Now we look at the side where we're seeing, all of our variables. So basically, what this program did is those 40 surveys that are out here on the left, we went ahead and put what we call the terminal node at 40 responses. So if we didn't have 40 or more responses, it didn't break anything else out. Because if we'd have broken out all 486 survey responses down to all 40 variables down to the last survey, this chart would have been taking the whole wall to put up. So we put a node split of no more. If it wasn't 40 or more surveys still included, it was out. So whatever else those guys were doing, if they plotted all that population, that survey wasn't included in the other side of the data. When we look at this side of the data, where our population is, that 67,500 seeds per hectare, the next most important thing was rating. Where we went, if the rating was, I think we put 1 as our. We put, excuse me, I gotta think a little bit. It's been a little bit long for me. Yeah, right. So we must have went with the ratings. So the rating of 5.2, basically we put our ratings on a scale of 1 to 9, saying 1 being the most susceptible, and 9 being the most or the most, or would have the most disease. So basically at a rating of less than 5.2, 15 surveys out of 115 tested negative. Going the other way, if the rating was greater than 5.2, 96 out of 220 were negative. So about 40%, on the left there were negative versus right there about 18% on the right. So kind of showed that rating was really a big deal, and that was a big part of, I think, why we have seen a decrease in Gosses, is because people have really been watching those ratings. The third one variable we saw was longitude. Rough estimate, that's kind of Missouri River between Nebraska and Iowa. So if you're looking at the longitude there, if you were, kind of stayed on the Iowa side of the border there, greater than 95.73, or no, that was the Nebraska side, excuse me, getting confused here, but the longitude of less than 95.73 would have been the Iowa side. So right at about half the surveys were positive, half were negative. When we got over to the, where a person was applying a susceptible product, a rating of greater than 5.2 at a longitude of greater than 95.73, which would have been basically the Missouri River into Nebraska, applying a simple product, only 3 out of 48 were negative for Goss's wilt. So you're looking at almost, 9% or not even 6% being negative, 94% positive if they use a susceptible product basically in Nebraska at that point on those surveys. So it just kind of showed that, the disease came from there, probably still has a strong hold there, and some of that made a big difference. If we go to the random forest, so like I said, the problem with this CART program is every time we run it, now if we did our node split at 40, we would probably get almost the same result every time we ran this program. If we just went down lower, when you get down to those lower numbers, all of a sudden that program's going to change every time we do it. So what we used the random forest for was we ran this, and all random forest does is it basically took that tree I had in the first graphic and it ran 2,000 times and averaged them, and it says, this is the predictor. So when we look at this, in the CART model, plant population was our overarching highest factor for Goss's wilt development. In the random forest, we're saying Goss's wilt rating, or what the seed company's rating was the highest development. And then what we did is we wanted to kind of rerun this. Let's take out all those variables that are fairly low on our Gini measure chart. Let's just look at these top 14 variables. We kind of got a break there. Let's look at these top 14 variables and see how that data comes out. And so it's kind of a close-up so you can see them a little bit better. But Goss's wilt rating, population, rotation, again being residue borne, continuous corn's not going to be good for it. Planting date did come up in there, and then percent residue covered. All the way down there. It kind of pushed the longitude quite a ways down the list on this one, where it came up more on the first one. But when we went ahead and reran just these 14 variables from random forest, just put the 14 back into our CART, really got very similar things what we did the first time. So it was a interesting thing. I think the thing we learned from it, as kind of those ratings, we've been preaching these ratings are what guys need to follow. I think the guys have started to follow the ratings on that a little bit better. Not real sure why the planting date came in there. I don't know that anybody started to work on any of that research yet. Then we get to the rotation. That one seemed to be pretty big again, where if we had some sort of rotation down there towards the bottom, 47 negatives to 39 positives. Versus in a continuous corn, 18 negatives to 43 positives. So a lot of this is probably a management thing, which we kind of knew going in. Like I said, it was kind of to help lead some new research on the pathogen. And as I was telling Jim here earlier too, like I said, this pathogen really kind of went away, and I think they're focusing a lot more of their research and efforts right now on the bacterial leaf streak in corn. So since my project, it's kind of died back a little bit, but I guess kind of in summary on that aspect of it, we found several factors that influence Gosses. Disease prevalence has been very low since 2011, and I think that's due to people were rotating crops a little bit better. We're seeing soybeans further west than we did, people getting out of corn, and I think we're getting better genetics, better genetics that are helping, where guys are more conscious of it. They know we need to plant something resistant to the pathogen to help out. So I guess with that, does anybody have any questions on any of that?